continuing with siRNA importance, that is siRNA in the treatment of asthma. So siRNA based technology targeting the transcription factor, that is GATA3. GATA3 is mainly responsible for the overexpression of what we say T helper 2 cells. So by using GATA3, we are going to silence T helper 2 cell that will be helpful in the treatment of asthma. So before understanding how siRNA is going to help in the treatment of asthma, we are going to pathogenesis of asthma. That is, we are going to mainly see two types of cells, that is stratified squamous epithelial cells and goblet cells. These goblet cells is mainly responsible for the production of mucus. So one say that allergen comes and binds in this path, and once it is detected by the stratified columnar epithelial cells, it will be the information will be passed on to dendritic cells. These dendritic cells will be synthesizing or synthesizing certain cellular signaling molecules that will be acting on goblet cells. These goblet cells will be uh, acting as a defense mechanism by producing more and more mucus. And there will be overproduction of mucus in this layer. So in the pathogens of asthma, we can also see that there will be increasing content of eosinophils in this layer. So once there is a form, there will be release of chemokines through the synthesis of thymostromal lymphocyte protein. So once chemokines is formed, it will be activating neutrophils, and from neutrophil, there will be formation of more and more T helper 2 cells. So once there is formation of T helper 2 cells in the bronchioles, it will be mediating its function through the various interleukins. So we can see here how T helper 2 cells plays a main role in the pathogenesis of asthma. By with the help of interleukin 5, it will be going to increase in the eosinophil count. That is, we can see here there will be more and more production of mucus and eosinophil. There will be more and more thickening of the mucus. There will be gas exchange will become very difficult in this interleukin 5 through the mediation of interleukin 5. By the mediation of interleukin 4 and 13, it will be going to stimulate the B cell. This B cell will be stimulating immunoglobulin E antibody. It will be in immunoglobulin E antibody. It will be binding on the surface of muscle. It will be going to release the histamine. As we know that histamine is responsible for the contraction of bronchioles. So that is of course the pathogenesis of asthma. So T helper 2 cells by the mediation of interleukin 9, it will be going to act directly on the muscle, releasing histamine. So ultimately we can see that T helper 2 cells is the main plays a main pathological role in the asthma. So with the help of GATA3, GATA3 GATA3 gene is nothing but the gene which is going to control the synthesis of Th2 cells. So by using siRNA technique, we are going to silence the gene, that is GATA3, so that will be helpful in the treatment of asthma. siRNA activity in dendritic cells. So as we know that in previous slides we saw that dendritic cells plays a main role in immunity. That is it will be binding to toll-like receptors and initiate the um, antiviral response, or it will be activating the immune system by binding to toll-like receptors. So some researchers has found that CD44 gene is responsible for overactivity of immune system. So overactivity of immune system is found in autoimmune disorders. So by using siRNA technique, we are going to silence the CD44 gene that will be helpful in the treatment of autoimmune disorders. So how siRNA is useful in the treatment of autoimmune disorders. So encapsulated siRNA. As we saw in the properties that siRNA is negatively charged, it is going to coat with positively charged proteins or lipids. That coated siRNA is incorporated into the dendritic cells. So when there is overactivity of dendritic cells, this coated siRNA will be going to silence the CD40 gene present inside the dendritic cells. So that will be helpful in treating autoimmune disorders. So as we saw in the approaches how to improve the characteristics of siRNA, that is using different formations such as cationic liposomes or polymers. Encapsulation method we already saw. Liposomal and polymeric nanoparticles coated with targeted ligands have been found useful in delivery. Thus, a combination of these methods to generate stabilized siRNA encapsulated nanoparticles may provide a method of choice to enhance delivery. siRNA in neurodegenerative diseases. Specific siRNA knockdown of mutant SOD1 gene also shows amyotrophic lateral sclerosis. This disease mainly affects the motor function. So there will be over expression of SOD1 gene in case of amyotrophic lateral sclerosis. So by using siRNA technique, we are going to knock down this gene, that is over expression of SOD1 gene. So that will be helpful in the treatment of amyotrophic lateral sclerosis. So as we know that Huntington disease is nothing but involuntary movements. There will be no control of movements in an individual. So there will be dance-like movements. Irrespective of the person, there will be no control over the voluntary movements in case of Huntington disease. It is also affects the what we say yes motor function or brain function. Everything will be affected in case of Huntington disease. So by using as we see into the Huntington disease pathology, so there will be CAG repeats. In normal individual there will be 15 to 25 CAG repeats, but in case of Huntington disease person, there will be CAG. There will be approximately more than 35 to 40 repeats. So there will be overexpression of that particular CAG repeats. So by using the siRNA technique, we are going to knock down that gene which is going to code for CAG repeats. So there will be less expression of that gene. By using siRNA technique, we are going to knock down that gene. So that will be helpful in the treatment of Huntington disease. So miRNA has nothing but it is nucleotide length is similar to siRNA, that is 21 to 24 nucleotide endogenous initiated non-coding RNA that post transcriptionally control gene expression by either translational repression or mRNA degradation. mRNA they play a significant role in regulating mechanism operating in various organisms, including development, timing, and host pathogen interaction, as well as cell differentiation, proliferation, apoptosis, and tumorigenesis. miRNA pathway is similar to siRNA. So in miRNA, we can see if there is a pre-mRNA. How pre-miRNA is found? That pre-mRNA means primary miRNA is found. So it will be looking like this. How it will be formed means there will be activation of RNA type 3 endonucleases that mainly based on complementary. It will be in a single strand there will be arrangement like this, that will be formation of primary miRNA. So primary miRNA nucleotide length will be between 100 to 120. So by using the Drosa enzyme or DGCR8, we are going to cut down certain parts in primary miRNA. There will be formation of preliminary miRNA. So this preliminary miRNA nucleotide length is between 65 to 70. So once pre-miRNA is formed, that will be transported from nucleus to cytoplasm by the formation of nuclear pore complex by using the enzyme exportin 5. So once it is sent out of the nucleus, that is into the cytoplasm, there will be activation of dicer enzyme that will be cleaving the preliminary miRNA. There will be formation of activated miRNA. So there will be formation of overhangs 5 prime 3 prime overhangs in the cytoplasm. So once there is a formation of this overhangs, there will be binding of risk complex as we saw in siRNA. The rest of the mechanism is same as what we saw in siRNA mechanism. There will be attachment of risk complex to this component, and there will be degradation of this strand, that is passenger strand and guide strand. Guide strand will be attached to the H complex, and this passenger strand will be released into the surrounding medium. And we know that passenger strand is non-functional. Guide strand is helpful in identifying the malfunctioning mRNA. And once malfunctioning mRNA is founded by this guide strand by using RISC complex, that is, it will be containing certain proteins and organoid protein and certain components. It will be responsible for slicing the malfunctioning mRNA. So we are going to importance of miRNA, that is cleavage of HIV1 proteins by miRNA. We know that miRNA is known to replace mRNA translation through recognition of specific binding sites usually located in their 3' untranslated region, thereby inhibiting the translational inhibition. This anti-HIV therapy is one of the RNA-based strategies that include antisense, ribosomes, and aptamers. Some of these are currently being tested in clinical trials. So this technique is not available now at present. Uh, uh, just we are going into the theoretical aspects of siRNA and miRNA. So as we know that in case of AIDS, that is which is caused by HIV1 gene, HIV1 virus, there will be incorporation of genetic material of this virus into the human genome. So we can see in this diagram, there is incorporation of HIV material. So by using once there is a immune system activation, there will be overexpression of this CD4 cells. So once there is overexpression, there will be over multiplication of this. There will be spreading of this HIV component to various cells. So by once it is identified at initial stages, we are going to silence this part, this gene, by using miRNA technology. So next is miRNA targeted glioma associated antigen 1 protein. So more than half of the human genome are located at sites known to be involved in cancers. mRNA is mainly helpful in identifying the cancers such as phagocytes, minimum regions of loss of heterozygosity, minimal regions of amplification of common breakpoint regions. We engineered glioma 1 miRNA. This glioma 1 miRNA is responsible for inhibiting this SHH pathway to downregulate protein expression. We know that SHH pathway is known head-Georg signaling pathway. This signaling pathway is mainly responsible for cellular differentiation, cell processing, and over multiplication of over multiplication of the particular cell. There will be over activation of this pathway in case of cancers. So by using GLI1 GLI1 miRNA, we are going to silence certain components that will be suppressing SHH pathway, that will be helpful in the treatment of course, cancer. miRNA in lung cancer detection. We designed a sensor based on system and kept gold nanoparticles that makes it possible to detect miRNA related to lung cancer. So a single standard probe, SS probe, was covalently immobilized on the surface of system by glutaraldehyde solution. This immobilization is carried out by using glutaraldehyde solution. And then the SS probe was hybridized with the help of lung cancer specific miRNA. So the preparation process of this proposed electrochemical nanosensor was characterized by scanning this single immobilized ssRNA SS probe was hybridized with the lung cancer specific miRNA. This hybridization part, it will be different from that of the miRNA which is found in cancer cells. So by using this difference, we can found out the lung cancer. So the preparation process of the proposed electrochemical nanogeneration was characterized by scanning electron microscopy, cyclic photometry, and electrochemical impedance spectroscopy. So by using this technique, we are going to find the difference between hybridized mRNA and the mRNA which is already present in lung cancer cells.